So Maria Jose Clavijo
Yes, great.
is a half-time research associate professor in the Veterinary Diagnostic and Production Animal Science Medicine Department at Iowa State University. Her research focuses on clinical and field-based investigations of infectious diseases in livestock, with particular emphasis on bacterial diseases of swine. She leads projects involving the use of next-generation sequencing to better understand pathogen dynamics and support disease prevention strategies. Prior to focusing exclusively on research, Dr. Clavijo spent 10 years working in health assurance at PIC, where she designed and implemented whole herd disease prevention and elimination programs across multiple countries while concurrently holding her position at Iowa State. She obtained her PhD from University of Minnesota in 2014, studying the epidemiology of Mycoplasma hyrinus in U.S. swine production systems. Dr. Clavijo earned her veterinary medicine degree from the University Central de Venezuela in 2009, where she also practiced before pursuing her PhD.
Very good. Thank you so much, Caitlin. I appreciate it. And I would like to start off by thanking everybody today for attending. Ooh, okay, can't do that. Maybe turn it, yeah. Thank you, appreciate that. No, this is too loud. Okay, can you hear me well? Yeah, yes, okay. Well, I appreciate also the organizing committee, Dr. Rodemaker, Dr. Greiner, and Stacy for organizing and putting these types of events together. For a person in academia, it's incredibly valuable to have these spaces where we as researchers can understand your pain points, your areas of need, so we can make more of that applied science that helps you in your everyday. So I, again, really appreciate these spaces of interaction. I'll start off with this. When we think about high health production, it was really up to very recently where we thought of these as, well, just the top of the pyramid, or very niche, or luxury markets, right? Think of ABF production. But I think the game has changed. And I think that given all of the constraints in today's global market of labor challenges, focus on more sustainability, regulations on antimicrobial use, labor challenges, I think I already said that, and consumer expectations, what we're thinking and what we're seeing is that high health pigs today is a competitive necessity. So what's chipping away at that margin, or what's chipping away at our production, right? And it's very important for me. I am a bacteriologist, an epidemiologist. I tend to look into endemic diseases, bacterial endemic diseases. And when we think about these types of bugs, we have to think about the epidemiological triad. So you have your pig, your environment, and your pathogen. And it's the unbalance of one of these that leads to disease. So once you think through this picture, you start to think about, well, controlling endemic bacterial agents doesn't necessarily mean just controlling the bug. It's also about controlling the environment and those pressures. It's also about understanding the pathogen and its qualities and its ability to cause disease. But it's also understanding the pig and that potential immunosuppression or not, or the immune capacity that they might have. Because at the end of the day, for all of these bugs, we need to increase immunity and decrease challenge. So let's talk dollars, right? You see one of the most impactful agents in pig production, the bacterial agents, not necessarily the viral, I left those out. But this table highlights what key endemic bacterial diseases are costing us. So anywhere from APP to E. coli, daily these are chipping away. These are not, rare outbreaks. We're experimenting all of these in a single flow, in a single pig, every single day. And with the right strategy and with the right approach, and again, creating these environments where we understand how we can develop tools, understand the bugs, and find and develop solutions for them, I think where we can start to address these issues. I come from the D Lab. It's the largest veterinary diagnostic lab in porcine in the world. It's 125,000. What we have here is a true gem for the world, okay? I live in Spain, I've lived in Mexico, and in both of these places, they see Iowa and they look to the diagnostic lab to understand the trends, the tools, and the bugs. So again, we have an amazing asset right here. So in working there, we wanted to understand what's the burden of disease for these endemic agents. And the bottom line is that we saw a significant increase of all of these agents: Streptococcus suis, Mycoplasma hyarinus, Actinobacillus suis, and Glasarella parasuis. So let's highlight here what we saw for strep suis. 1 in 5 pigs diagnosed at Iowa State has strep suis, okay? We'll talk about a little bit why that has changed, that picture has changed, that significant increase that we've seen over time. But the bottom line is, we normally just think of PRRS, of PED, of these bigger mycoplasma, more impactful diseases. But think about, again, these bugs each chipping away at us every day, that cumulative, they take a big chunk out of our margins or productivity. And what we saw was that, again, these bugs cause different sets of diseases, and we were seeing that each disease, so not just meningitis or endocarditis, they were causing a lot of different diseases, and that was being increased over time. We also saw how, depending on the type of disease that strep would cause, you would see it in different parts of the production chain, or their growth. So this is not strep suis. It's not just a bug of post-weaning or pre or peri-weaning stage. You're looking at an impact across the board. When we look at Glasarella, again, significant increase in 5 years in the diagnosis. Guys, this is not detection. This is diagnosis. This is a pathologist looking at the case and saying, this pig died of this disease. This is not just, oh, I detected these endemic bugs in the lung and that therefore. So again, a lot of bronchopneumonia caused by Glasarella and a lot of serositis. So that thickened pleura that you see, those cheesy hearts and lungs that you see when you open up pigs. One of the biggest ones that increased was A. suis. And it was very interesting to see this bug pop up. Again, 8.7% of all sepsis cases were A. suis, and we saw a lot of increase in serositis. So we don't talk a whole lot about A. suis. We truly don't know a whole lot about its disease ecology. So, but you see the results, and it is, significant, causing significant disease. And what are we doing about it? And then the bug that, when I started studying hyarinus in 2010, most people would just say you're making up diseases to justify a PhD. And what we ended up proving was that hyarinus was associated with, although this had been published for decades, we just didn't listen. And we now know it's a significant cause of arthritis and polycerositis in pigs. So, and the data shows that, right? We saw a significant increase of arthritis and polycerositis. The other thing is that there's differences in strain, so you see a lot more of that impact depending on the strain that it is. One bug that was very interesting for us was Hyacinoe. Now, if I wouldn't have seen the data and I would have hypothesized what we would see, and I would have thought, no, we saw a massive increase of Hyacinoe, and then it actually decreased. And I'm sure it's not because you guys don't have lameness cases in your farms. I bet you would probably complain about post-weaning or growth finish arthritis, but we don't get a whole lot of cases from them. And there's a reason why. Diagnosing lameness is one of the most frustrating things that a pathologist does, because we never can pretty much pinpoint. There's so many drivers and risk factors and causes that it's difficult to diagnose. So we attributed this to just a lack of cases coming in, or it's an agent that causes disease in the grow finish. You have an animal that you can treat, you're not gonna send it in for necropsy, right? It's a more expensive animal, it's harder to do a necropsy, to send the samples. So we think it's still a very important agent that we need to better understand what's it doing in pigs. But then you can't forget about these endemic agents and that, that triad, okay? So you have other coviral agents that are in the same pigs running around or causing and creating these synergies and worsening the picture. So while we saw most of the disease being diagnosed alone, a lot of these cases were co-diagnosed with PRRS and flu. So another way of saying it is that if we don't get ahead of the viral diseases, we'll never get ahead of these endemic bugs, right? Alone, these bugs can cause disease, but when you add a viral component to it, it gets a lot worse. The other thing that we found, and this is the biggest take home for me, that I think we can say today, is where you collect your samples matters, okay? These are endemic agents. They live in the host, and some of them are varied in terms of their genetics, and they are commensal versus very pathogenic. The take home here needs to be, you need to collect the samples of where the disease is at. If you have a lameness, don't send a lung. If you have polycerositis, send your pleura. If you have meningitis, send CNS samples. What this is showing is pretty much if an isolate came from the lung, it had a less probability of diagnosing strep suis than if it came from any of the systemic sites that make sense for that bug, okay? So yes, we get a ton of strep suis from the brain, from the lungs, but that's not leading to diagnosis. So take home is, and I know it's very simple, but you wouldn't imagine the amount of cases that we get, and now I get in Spain is, I have a ton of polycerositis. Here you go with a nasal swab. Yeah, it's going to be in the nasal cavity. It's going to be in oral fluids because these are commensal agents. But the strains that are causing disease in that tissue, in that systemic tissue, is going to be different. So again, with the stakeholders, with your clients, with your producers, think about what's your objective when you take samples. Are you trying to understand what's causing clinical signs today? Are you trying to get an MIC to justify some control programs? Are you trying to understand what's the role of this particular pathogen in my herd? Or are you trying to create a diagnosis? This is one of the most common things that I see is I'm trying to get an isolate to build an autogenous product. So think about that objective. The next thing that I can tell, if you can share this with your clients as well, is, or yourselves if you have farms, is to, well, I want to take samples, what's the best pig? The amount of times that I see this pig being submitted to, at least in Spain, where I'm at, is a lot. You're not going to get anything reasonable from these animals. You're going to get probably isolates that are not causing any issues, or you're not going to get anything. So send in pigs with fever. That's the basic. Bring in your thermometer, take samples. If a pig has fever and you're trying to target systemic agents, look for dyspnea, that lack of being able to breathe, lameness, paddling, if you're seeing any of the CNS or clinical or central nervous system signs. Don't take any treated pigs. Antibiotics are trying to grow the bug to get you that isolate. That's not going to help. And then it takes time, for some of these flows to get the strains that we want. And to understand or diagnose, it takes 2 or 3 pigs per flow to figure it out, or maybe 2 or 3 times to visit the farm. So one of the things that I want to highlight here is the group, the SMEC group at Iowa State. They do a fantastic job of teaching and educating our next generation, veterinarians, students, and allied industry. So This is one of the infographics that we've created with them. It's to help, get these into the SLAT level to understand how you can better take samples, better get those agents out of these farms. So again, this is a video, how to collect samples. This is on the website, on our Iowa State VDL website for you to see. We also created these infographics, and we're about to produce some more videos and these types of educational materials. Again, the biggest take home from that paper that we did was we're not taking the right samples. We're getting lost in that mix, and we need to improve that. So again, kind of the things that we look for is for strep suis, taking that whole head and just sending it in. No need to take meningeal swabs. Anything that needs to get done at the farm is prone to contamination. So just best to send whole legs, whole pigs, whole heads, rather than opening up that pig and leading to contamination. Then we get the call, hey, I grew this E. coli from this plera. What, should I? No, that's probably contamination. So again, that type of thing is important to realize. Again, arthritis is one that I recommend just sending the whole leg. There's a paper again out of SMEC that showed that whole pig or the whole leg led to more diagnosis of the agent, or they found an etiology just by sending in that type of sample instead of opening up the pig. This is hyorinus. So this is one of my pigs during my PhD. There's only hyorinus in these. There's no GPS or Glasarella. And this is Glasarella. And this is Glasarella. So you can see how similar these lesions look. And why it's important for us to tell those apart. Just a couple more pictures here just to show you. Sometimes people send us like very inflamed, hard joints, and those are just probably not in the articular space. They're filled with pus. It's probably some sort of trauma, but they look at this and they say, oh, strep suis. When you open this is just pus. You're not going to even grow anything out of that. So again, getting together with your veterinarian, your diagnostician, to understand what are the appropriate samples that you need to collect. And then the last thing, and I think you guys do here in America do a fantastic job at collecting the right set of samples, labeling them, putting them in coolers. It's impressive what I've learned in Spain about coolers and the lack of bags. So it's important that we, from each pig, collect different samples and put it in different bags. And we separate tissues. Do not mix lungs with anything else. Lungs, there is a chance that you'll get some tissue or some isolates that are not from systemic tissues into the other ones, and you'll confound diagnosis. So one of the things that I was, as I mentioned, how am I doing on time? You good? I worked for 10 years for PIC, and at the time in 2018, we had a lot of clients complain about strep suis. So what we wanted to do is create this partnership between Iowa State, the VDL, producers, and then we have several funding agencies that help put together a platform where we will integrate isolates, data from the clients, and the next generation sequencing technology like whole genome sequencing or bioinformatics. So this is not just an academic exercise. This is not just collecting bugs. This is collecting bugs with context. So flow, genetics, tissue type, histopathology, the age, and bringing that together to understand better what's the clinical relevance of these agents that we're diagnosing. So this is a database that ISU VDL continues to gather. We have a lot of different species that we look at, and this is a service. So you all can utilize this, or your clients can utilize this to characterize their endemic agents. So this is what it looks like. We do about 220 reports a year, and clients just send us their isolates and we put them into these trees. These trees are built with endemic strains from our regions, right, from North America. And we put in the strains that are most common, the most pathogenic, and they plug in their sequences into these trees. And then we're able to create a report for them that puts into context all of that and tells them, well, this is a bad one, autogenous vaccine, or this is not a strain that you need to worry. This is probably commensal or opportunistic. You can pretty much ignore that strain. Or, we say a lot, keep looking, because that's not the strain. And you will always get these conversations of, but it came from the brain. That isolate came from the brain, yeah. And we can contaminate the brain very easy. Strep suis is everywhere on that pig. And most of it is commensal strep. So it can easily get into your samples, and then you throw into the bottle the strain that's not causing disease. So with this program, you can do targeted interventions, so we can identify circulating strains, understand what strains are causing particular outbreaks, understand relevant antigens for vaccine, understand, for example, we get the whole resistome, so you can understand antimicrobial patterns. We can do pathogen surveillance. Pig flow management was the biggest. I talked about PIC and that story. That led PIC to take major decisions on what flows to keep and what flows we didn't want those to work with those flows anymore because they had strains that we could avoid. So depop, repop, and what we ended up seeing is back in 2017-14, the major strain in the US of strep suis was ST28. Let's call it baby strep suis because it wasn't that pathogenic. And we would make fun of Europeans complaining about strep suis. Well, in 2017 or so, we started to see an uptick in ST1. ST1 is the most pathogenic strep suis. And as you can see, that pretty much replaced ST28 in our database. But over time, what we've seen is that between NGS, mapping, and what I've just mentioned about some of these herds being depopped, and then widespread use of autogenous vaccines, we've seen over time, we barely see now ST1 being submitted from cases, okay? So you can see how we can start to now do strain level elimination. But you can also utilize this technology to just what I just said. Well, you know what? My farm has source A of serotype and this is source B. I'm not going to mix these two flows, right? And we saw that quite a bit. The other thing we can do is now we're getting to a point where we have hundreds of isolates for our production system. And what we do is create a particular tree for them. And they start to gather over time particular strains from their herd and are able to make targeted or focused or focus on which strains to combat. Again, another example, we found this emerging ST454 strain here in North America, a Glasarella parasuis strain that caused a lot of disease. And what, again, ended up happening was, well, we really don't want to live with those strains. We're getting to a point where we have systems in the US that if they're naive for PRRS and they have a bad strep, if they break with PRRS, some will decide to depop just to get rid of particular strep suis strains. And we're moving on to E. coli, same thing. Now I get to share some of the data from Spain. We see highly resistant strains in Spain, but not very toxigenic. And then you see somewhat resistant strains in the US, but highly toxigenic. So now you see global differences. So one size fits all approach won't work for these bugs. We need to understand the ecology at different levels. So then I wanted to go into autogenous vaccines because being in PIC, one of the biggest things is I remember that really strep suis vaccines wasn't a strategy that a lot of our production systems used or our clients. And I think is what these types of tools have allowed, and really understanding the molecular epi, is that we know what are the antigens and the strains we need to put in that bottle. So for us, and it's our stance, is that if you get the right antigen, the right adjuvant, or a robust adjuvant, and the right amount of that antigen, these products are effective. They're not bulletproof. They're not perfect, but they're bacterians. But they, with a holistic approach with, medication and proper management, you will get a good benefit out of these. And then I share some examples of these are clients, we did some of this investigation with them about their strains and modified or developed their vaccine choices, and we got impressive results. And then lastly, we can track antimicrobial resistance with these bugs, with these tools. And again, this is just to highlight, and it's very counterintuitive, but you're looking at Europe, Latin America, and North America for strep suis. And you're looking at 3 big buckets: the pathogenic, the potentially commensal, and the potentially pathogenic. This is all done via a bioinformatics tool that we've developed with a colleague in Wageningen University. And the interesting thing here is you tend to see some global or regional differences. You see Europe with more resistance in the putative and the pathogenic strain. So you see a lot of these red dots means resistance. Less than what you see in North America, okay? And here's that stats for you. But the interesting point was the resistance is being harbored in the non-pathogenic strains. So what ended up happening before is that we would submit an isolate, you would get a highly resistant strep, put it in the bottle because it's probably highly resistant. That's probably the one that we should focus on. And it's actually, we should learn that no, the ones that are highly resistant are not the ones causing disease. So just how am I with time? 10 minutes, okay. So just high level to wrap up, again, I'm more focused recently with strep suis. We recommend treatment. There's 2 levels, right? You have disease, yes. Big thing would be, or an emergency treatment would be to do IM Ceft or enrofloxacin and some Dex. You could also treat these pigs with penicillin and amoxi in the water. And then more of a preventative, again, you can do this here, not in Spain, but IM Ceft to all suckling pigs ahead of first clinical signs. And on the diagnostic sides, it's that piece, what you want to know, what's the bug causing disease in your herd. And then what I said, right, it's that other part, the immunity challenge balance. You want to maximize immunity and minimize challenge. So you want to do, people just do gilt autogenous vaccines, but also pre-faro. Very important to assure colostrum intake. And then on the minimized challenge, all in, all out, this is in Spanish, but all in, all out, you need to control your viral diseases. And then now more in Spain, you're seeing more of that batch farrowing because of the heavy endemic pressure that they have and the smaller herds, they can get away with a 5-week batch. And that's been very effective for PRRS, E. coli, and all these bacteria agents. Reducing stressors, again, those high temperature changes or high humidity are very are risk factors for these bugs. And then so that database, and getting to a little bit more of science before I conclude, that database fed into this big project that we did at the global level with these experts in strep suis. They were able to pinpoint a gene in the pathogenic lineages of strep suis. And from that we developed a PCR. So this is taking not basic science, but pretty high level science down to the field. So we took that and we developed a PCR, and we wanted to see not only where does strep suis live. Again, strep suis is one of the most common species in pigs. It's the most common in the oral cavity. It's where do the pathogenic streps live? So we developed 2 PCRs, one that targets all strep suis and one that targets only the pathogenic ones. And this was done by my student, Dr. Robert Mugabe. And what we wanted to understand was the dynamics of infection at the lactation level. Sow and piglet in that relationship, but then the temporal changes, and then that weaning stage. And what high-level view, what we saw, gilts are more likely to harbor disease-associated streps, okay? Not only that, is that yes, strep is in the environment of the sow, but not at the levels that we thought. And also, now we're finding it in samples that we didn't know they were at but essentially positive piglets that came from sows with udders that were positive for disease-associated strep were 13 times more likely to have disease-associated strep. Not the disease, just the bug on it in the oral cavity, okay? So this is just looking at antemortem samples, seeing where does strep suis and how does it transmit and where does it hide, let's say. And then we look at the piglet. So this is a flow chart. So you see the 3 time points that we took on piglets, and these, every single one of these is a pig, and you can see the trends there. First of all, this is farm 1 and farm 2. So differences, red is positive, blue is negative. So again, differences between farms, but also what was very impressive is that we are weaning a high number of disease-associated strep suis negative pigs. So when we thought, oh, most pigs have strep and most pigs have pathogenic strep, that's not true. We're weaning a big number of negative pigs. So if you combine those two things, we know that gilts have a lot of strep, and we know that we're not necessarily weaning a whole lot. I think there's chances for some, programs that can at least reduce the disease burden of these agents, maybe potentially eliminate. That's probably something to consider in the future. But again, if we don't have the tools to study the bugs, where do we begin, right? So that's why it's important that we do this type of endemic work. And then just wrap up. Endemic agents remain a top health productivity challenge. To study it and to understand it and to control it, you need to get to the right pig, the right time, and the right tissue. Not all isolates are equal, so we need to have these newer genomic tools to understand and differentiate them. For us, autogenous vaccines are useful but not universal. You need, and it requires a multifaceted strategy: gilt acclimation, smarter vaccination, responsible antimicrobial use, better pig flow and biosecurity, and then integrate that with your genomic data and surveillance. And then finally, we need to educate and train more our farm personnel and create that interdisciplinary collaboration of vet nutritionists, geneticists to address these issues. And with that, I would like to thank you for your time and thank you for the invitation. I would entertain any questions.
Are there any questions for Dr. Clavijo?
That are not Brian. I'm kidding.
Yeah, Maria, great presentation. With some of your learnings over the last few years, has it changed your approach or focus on gilt entry, biofeedback in the systems that can do that, or even parity structure on farms, more closures and things of that nature?
Yeah, or parity seg?
Yeah.
Yeah. No, great question. And I think we really don't have much other tools for gilt acclimation, right? When we think about mixing populations or gilts going into farms with a different endemic profile, I mean, there's still 2 options that we could use. Understand how we expose them, create that immune and bring up that immunity in them. That's either through feedback, because some bugs we just don't have, right, the products to, or autogenous vaccines. We're seeing a lot more of that. And then the second part would be cool down, right? You need to have that time and that probably those products or medication to reduce that shedding. So we're not necessarily exposing or having too much shedding into pigs. So I think that's on the gilt side of introduction. But then on the, I think there's opportunities that we do more targeted management on gilts. We know that they produce less less good colostrum, and we know that they're shedding more. So there's definitely strategies. I don't know how that all combines with the efficiency of the production systems that we run, but one of the questions that we did for this paper is, do we need parity segregation back, and think about that to get rid of some of these agents. So yeah, great point, great question.
Congratulations.
Thank you,
Doctor.
I appreciate it.
Great presentation. Do you consider important the presence of strep suis in the vagina of sows as a source of infection for the piglets?
Great question. So in one farm, we saw that it was significantly associated with piglets having disease-associated streps. Again, every single pig that we sampled had the species, every single sample. But pathogenic streps in the vaginal canal or area, 13%, about, I think, between 13 and 20. I can go back to the slides, but not a whole lot of gilts had pathogenic streps in the vaginal canal. But in one of the farms, we did see that that was associated with them being positive. So again, that would be a great thing to, if we want to reduce disease burden, do we go ahead and do some sort of lavage, which I know has been tested for strep suis. On the other end, you end up thinking, well, but now you're killing are you potentially killing all of the commensal streps? And is that providing any benefit to the pigs? So, yeah. Thank you. He's the reason why I'm in pigs, by the way, pig disease. So he was my professor in Venezuela and my mentor. So it's very exciting for me to have him here.
I'm currently using an autogenous vaccine for strep on my farm. How often should I be checking?
Oh, great question.
To see if I need to switch or do something different with that vaccine?
If you're switching sources, genetic sources or gilt sources, if you have an outbreak, right? You have an outbreak, you want to check and then what the third one would be, do it at least, if none of these things happen, at least once a year, check on your strains. Anecdotally, what we end up happening is that you control some strains and then others pop up, right? It's logical. So then I wouldn't still, I would update it. I think Marcelo Gotcha has shown that you can put up to 5 antigens and be effective. Again, I don't have that experience, but it's what science says. So if you're not hitting those 5, I wouldn't replace strains. Strep suis persists in populations, and it doesn't change a lot over time. So you won't get rid necessarily of the strain in your farm. You might reduce disease, but you'll still have it. So if you have a PRRS break, a flu break, and you don't have that protection, you might. So I would rather add than remove, and you start to monitor that. So once a year at least, and then in outbreaks or if you're changing gilt sources.
This will be the last question.
Okay.
Yeah, thanks, Marie. So that prompted me to think about this. We don't normally think about bacteria as causing outbreaks, right? You said if we have an outbreak, you mean a PRRS outbreak, EDE outbreak, or something else? I'm not sure what you meant by that, but my question really is, what do we need to really change our mindset about that, right? When we think about biosecurity today, it's mainly for PRRS virus, PED virus, things like that, but are we moving some of these bacteria around too and don't know what the consequences are of doing that? We're not monitoring very closely. And
Right.
I mean, I think we're a ways from that, but I mean, it's maybe something to think about.
No, absolutely. No, good question. So on your outbreaks, it's just an increase in strep suis cases that's not normal for the farm. It wouldn't yeah. And for that is probably like 0.6. I don't know. It's very minimal, but it's something that prompts the farm manager or the veterinarian to say these cases are increasing. And your other question, yes. So one of the things that I never expected to see when we started to map out the multiplication system was how consistent you get the same endemic profile over time, and that doesn't shift. If you have proper biosecurity, we are not moving strains of strep, Glasarella around as you would think. So I think our biosecurity, our basic biosecurity is probably protecting us against. Not saying that it doesn't happen. We did have a couple of customers where we know that it was not possible for that customer to have that strain, and realized that through transport events, they had brought in the bad strep into the flow. So
Most of the time it's being moved with the animal.
oh, correct. Most time it's moving, and not only that, they won't get replaced. People think, oh, gilt acclimate, or bringing new gilts, I'll now have whatever genetics has. No, you'll have what you have, and the next genetics, and the following, and the following. So they don't go away. Thank you.
Let's make our speaker.
Thank you.